Today we will be looking at the technologies behind transhumanism and the progress that has been made in the field of human enhancement. If you are unaware of what transhumanism is, please go watch my previous video on the topic first. Now, at its core, transhumanism is built off of fields and new technologies such as gene editing and CRISPR, 3D bioprinting, biomechatronics, nanobiotechnology, and neuroscience. CRISPR are segments of prokaryotic DNA containing short repetitions of base sequences that are used in gene editing by delivering a protein called Cas9 and appropriate guide RNAs into a cell. By doing this, the organism's genome can be cut at any desired location. The simplicity of CRISPR, with only 3 required components, makes this system highly desirable for genome editing. CRISPR is more efficient and low cost than any other genome technique available. This potential was realized in 2012 by the Doudna and Charpentier labs. CRISPR could potentially be used to correct a wide variety of genetic diseases and disabilities, as well as increase our intelligence, strength, agility, and other genetically determined attributes. It can even be used to increase lifespan. Last year, using their own experimental gene therapies, US startup BioViva successfully reversed 20 years worth of telomere shortening in a human. Telomeres are short segments of DNA which cap the ends of every chromosome, acting as buffers against wear and tear. They shorten with every cell division, eventually getting too short to protect the chromosome, causing the cell to malfunction and the body to age. So by lengthening them, you are effectively reversing aging to some extent. Through genetic engineering research on animals, we have discovered many techniques that might enhance human abilities. At Princeton University, researchers looking for ways to stave off Alzheimer's disease have created mice who learn things more than twice as fast as their peers. Southern Illinois University engineered mice to live twice as long as their genetically normal counterparts by just changing a single gene. In a lab at the University of Pennsylvania, researchers trying to find a cure for muscular dystrophy have produced mice that are super strong. They can climb things and lift things that their genetically normal siblings can't, all just from being injected with extra copies of a gene for building muscle. That same gene also plays a role in human muscle development. At Duke University, scientists looking for ways to help amputees and paralytics have implanted a group of monkeys with electrodes in their brains. The monkeys can move mechanical arms just by thinking about it, as if their limbs were part of their own bodies. In Lisbon, Portugal, there's a group of blind men and women who can see. They wear cameras connected to electrodes implanted in the visual parts of their brains. Some of them were blind for more than 20 years before the surgery. The same research that gives them sight could beam images from one person's mind into another, like a rather basic form of telepathy. All these discoveries show that by unraveling how our minds and bodies function, biotechnology could give us the power to sculpt any aspect of ourselves. 3D bioprinting is another new technology with great potential. 3D bioprinting is the process of creating cell patterns in a confined space using 3D printing technologies, where cell function and viability are preserved within the printed construct. Bioprinting can be used to print bones, tissue, blood vessels, organs, and even scaffolds to regenerate joints and ligaments. If we are able to perform such feats, then not only will we be able to replace parts of our bodies with new healthier parts enabling us to live longer, but we will also be able to print parts that are stronger, more resistant, and more capable. We have already successfully managed to 3D print liver cells, esophagi, ears, tissue, bone, and even small-scale stomachs, lungs, kidneys, and hearts. Nanobiotechnology is a rapidly developing field with limitless potential. Some of the applications of nanobiotechnology include fluorescent biological labels, drug and gene delivery, biodetection of pathogens, detection of proteins, probing of DNA structure, purification of biological molecules in cells, tissue engineering, MRI contrast enhancement, and pharmacokinetic studies. Of most interest regarding transhumanism, however, is the possibilities of nanomachines or nanobots. These nanobots could be injected into oneself, acting as antibodies to protect against disease, extending our lifespans, increasing our physical capabilities, and even giving us entire new abilities and senses. Researchers say that one day in the near future, we will also be able to construct self-replicating nanobots that could assemble atoms into molecules, thus building new objects from the inside out. Not too long ago, at the University of Berkeley, a tiny implant the size of a grain of sand was created that can connect computers to the human body without the need for wires or batteries, opening up a plethora of endless possibilities and uses. The devices, referred to as neural dust, could be used to continually monitor organs like the heart in real time, and if they can be made even smaller, implanted into the brain to control robotic devices like prosthetic arms and legs. There are plans for neural dust to be used for chronic neural implants in the near future. It is currently tested by surgically implanting an array of them into an open skull. But researchers are aiming to get the size of the piezoelectric receivers down to 10 microns, so it can get past the blood-brain barrier as a drug. This was just a basic overview of some of the technologies and fields involved in transhumanism. My next video in the series will be a detailed look at human augmentation technologies and their future. If you enjoy these videos, please don't forget to like and subscribe. Thanks for listening and have a great day.